Hello, and welcome to this month's Simon Says Expert Series. I'm Dr. Rohan Maniar. I'm a medical oncologist, and I joined the Cancer Center almost 4 years ago. My research focuses on understanding and improving outcomes for patients treated with immunotherapy, with a particular focus in thoracic malignancies, including lung cancer, esophageal, and thymic tumors. I'm excited to have the director of the Cancer Center, Dr. Kelvin Lee, joining me today. In addition to leading the Cancer Center, he also has a research lab focused on multiple myeloma. Thanks for joining me, Dr. Lee.
Thank you for having me.
So before we dive into the remainder that we're tracking any questions you're asking live, I invite you to submit your questions in the comment box below or in the chat if you're joining us on Facebook. We'll do our best to answer the pre-submitted questions as well as any that come through over the next 45 minutes before we wrap up. So we're very excited to begin. Let's delve into our Simon Says topic for the month. Today we will be discussing Cancer Research 101, a focus on how ideas become clinical studies and all the important details and processes that occur as we take drugs from the bench to the bedside. Before we do that, I just want to get your first experience and what went into it the first time you went into a lab, Dr. Lee.
So, actually, the first time I went into a lab, I think I was 8. My dad was a microbiologist, and he worked in a lab. He ran a lab, and I remember he dragged me into the lab. He dragged me and my sister in the lab, and I was actually counting bacterial cultures. So there's this old-fashioned device that you put on. You put the agar plate on, and you have this little thing that if you touch the agar, it clicks. It clicks a button, and that's when that was my first memory of being in the lab. I would say the most important lab experience I had was actually as a high school student. I was actually going to become a marine biologist, and I had the opportunity to do a summer research program, which is why summer research for students is so important, at the National Naval Medical Institute under Dr. Elliot Perlin, who was actually a medical oncologist. And we were looking at the effect of, this was 10,000 years ago, we were looking at the effect of chemotherapy on immune cells, T and B cells. And we had really ancient technology to understand them. I won't even go into that because nobody will actually remember it unless you're 80. But that actually summer actually made me think, this medical research stuff is really interesting. So that's when I applied to medical schools and took me down a completely different path.
Yeah, I think likewise, I had a similar experience of getting into a lab, not necessarily finding it to be everything I was hoping it was, but gained a lot of valuable skills. And then ultimately later down the road, I found an opportunity where all those skills were useful, were very valuable. And then I fell in love with the laboratory experience and all that
Yep.
goes into those processes. So glad we have a very similar sort
Yes,
of
yes, there's
unique
a similar
experience.
path. Yes.
So I've always viewed cancer research as sort of three components to it: the basic science, the translational science, and then the clinical research. So Dr. Lee, for our viewers who may not be as familiar with cancer research, can you give us a sense of what some of those components are for cancer research and why they're so important to the field?
Yes. Well, let me take a step back and talk about what research is. I think that, a lot of people, especially that people that don't do research for a living, actually think of research as something mystical and mysterious. And the bottom line is that research is the process by which you use a structured process to solve a problem. So all of us have done this. I mean, the principles that all of us have gone through in life are the same principles as we use for scientific research. For example, let's say that your family's car is a 19 Honda Civic from 1987. And your daughter tells you that, hey, dad, mom, I got into travel soccer club, which means that we're going to be driving all over the Midwest to go to tournaments. And by the way, you're going to have to haul half of the team there. All right. So now you have a problem. I need a new car. And then you can break that problem down into pieces that are unique to your part of that problem. I need a new car that costs this amount of money. I need a new car that can hold 5 girls and all their soccer gear. I need a car that gets 31 miles a gallon. All right. So now you've you've actually made a more defined structure for the problem that you're trying to solve. And then you do what all scientists do. You go and see what other people have said about what those potential solutions are. You read reviews of cars, you go, and you talk to your friends and all that other stuff. And then you set up the experiment because, and the key piece of the experiment is that it has to have a measurable outcome. So then you go to the dealer and you test that car, right? You say, does it get 31 miles a gallon? Can you fit five girls and their luggage into the car? And you work all through that. So that is exactly pretty much the same way that we do science. And that whole structure of how we solve a problem, everybody on this call has done it in one fashion or another, solving the problems in their life. But science actually, so the structure in science actually, to Dr. Maniar's point, actually, we break it down into three parts, although all those parts actually all blend together. So we traditionally think of basic science. This is the fundamental biological principles that underlie why a cancer either behaves or doesn't behave in a particular way, or why your body responds in a particular way to a treatment or not. So these are usually, as we think about it, fundamental cell and molecular processes that typically will underlie both how your body normally works and also what goes wrong when you get cancer or any other kind of disease. And so this really forms the foundation of all of our understanding of all biology that goes forward. And I had a mentor, Craig Thompson, who was at Memorial Sloan Kettering, who actually simplified it for me way back when I was starting in the lab. He said, Kelvin, all biology is about food or sex. And essentially, we're studying the food part of it, but people study, where do cells get energy? How do they utilize it? Or how do they divide? How do they make more of themselves? Things like that. So that's where you start, and that's that core understanding. And that's why things like, all right, everybody makes fun of us studying fruit flies, right? How does fruit flies have anything to do with human beings? Well, they are what we call a model. It allows us to actually look at those fundamental pieces of biology in such a way that we can't do in humans. So, for example, actually, it turns out that the National Fruit Fly Library or Laboratory is down in Bloomington. They have all the different strains of fruit flies, and there's a strain of fruit flies that gets fat. It's an obese fruit fly. And there's another strain of fruit fly that gets pancreatic cancer. And so then the question is, if you cross the two, what happens, right? So you can do lots of interesting biology in very fundamental models that you wouldn't normally think have anything to do with human beings, but it really helps us understand the basic underlying mechanisms and processes that underlie all life. And then the second piece is now we've identified this whole constellation of processes and mechanisms and molecules and cells and all that other stuff. Are there things that are specific that get disrupted when something goes from a normal cell to a cancerous cell, right? So we define that, and then that allows us to pick out things that, hey, if I killed this molecule, the cancer would stop growing or dividing, right? Because this molecule is very important for that whole process. And then we move into the translational piece of saying, all right, with that knowledge, how can I get this into people, right? I've been working in fruit flies or petri dishes or whatever. How can I take that understanding and start to move it into people? And so that actually goes, do human beings do the same thing? Is it bad if you knock this process out? What happens to the normal cells? All those other things. What's the best way to get at that molecule? Is it another molecule to target that molecule? Is it an antibody? Is it an immune cell? The whole question of how do you get at that fundamental piece that you want to get at, that's all known as translational research, right? How do we move our science into our patients? And so once we begin to define that, and that actually has lots of different moving parts to it, in all the way from what's basic discovery, how do you target that molecule that you want to get at, all the way to how do you design the drug so it can be given to people in a safe or relatively safe way that's going to work. So that's that whole drug development process. And that's what you hear about when you hear about, big pharma developing a drug. That whole process is the translational piece. And then the final piece is for therapeutics, anyways, is this question of does it work in people, right? So you don't know, right? You've had these model fruit flies, mice, whatever. You have all these model organisms that you've tested it in. Things look promising, but people are different than all the models that we use in the lab. So then the question is, does it work in people? And that's when you do the clinical trials with phase one being, is the drug safe or is the therapeutic safe? What are the side effects? Phase two is, does it work? Does it kill the cancer like you anticipated it might be? And a phase three clinical trial is typically, does it work better than what we currently do now? And that's a comparison study. And so sort of traditionally, those are the three phases things have gone through. Some of the newer therapies have actually skipped the phase three piece and have just gotten to the phase two piece. And then the Food and Drug Administration reviews all that data and then decides whether or not you can actually sell it or market it as a drug for lung cancer, prostate cancer, multiple myeloma. So that's the whole process. And it is actually quite extensive, but it's getting faster and faster and faster. Now, the one piece of science we haven't talked about is actually population science. So population studies are larger questions of why do people do the things that they do? Why do people smoke? What are the best ways to get people to stop smoking? What are those barriers so there's the epidemiology and population science that is not traditionally thought of as sort of the basic clinical and or basic translational clinical studies, but are critically important for us to understand, how to manage cancer.
Yeah, so I think you gave such an excellent description of something that's sort of been maintained for, I mean, hundreds of years of science, which is the method, right? We ask a question, we research, we perturb when we can, and we see what happens, and then we validate our hypothesis or we reject the hypothesis, and we sort of go back to the beginning. It's an iterative process that we do
Right.
over again. But I guess for the last, let's say, 125 years of cancer research, things have changed. Maybe not
Yes.
so much in our method out, maybe not so much in the questions we're asking or how we're asking it, but the assays that we're using.
Yes.
So things started along really in like the early 20th century with Sid Farber in a
Yeah.
musty basement, looking at nitrogen mustard related toxins and seeing the effect on leukemias, lymphomas. As we fast forward over 30 or 40 years, we're talking about the emergence of molecular biology. So since those days of Sid Farber in a musty lab, what does the research process look like now, and how has it evolved since then?
It is actually quite spectacular, and it's evolving on any number of fronts. It's evolving because of new technological breakthroughs in the way that we think about things. It is evolving because AI is becoming involved in how we understand data. Our ability to really interpret data is primarily based on how much data we can actually analyze all at once. And AI has increased that logarithmically to the point where, the enormous amounts of data that no human or no team of humans could actually put together, sit down and synthesize. AI is being able to do that. And our ability, that is tied very closely with our ability to generate enormous amounts of data with, things like genetic sequencing, for example, now gives us an enormous pile of data to work through. And really, it's gotten to the point where AI is the only way to actually sort of make connections between data simply because AI is the only way to get that all under one roof. And also, the advances have actually happened because we understand the fundamental processes better. So, a perfect example is actually the immunotherapies called checkpoint inhibitors. So these were drugs that actually block how tumors shut down T cells or immune cells. And by using these checkpoint inhibitors, you unleash the T cell and then allow it to go kill the cancer. And this was really, I would say, one of the fundamental breakthroughs on the clinical side that brought cancer immunotherapy into the forefront. So drugs like the drug that Jimmy Carter got that cured his brain melanoma, and all those drugs that you see on TV now for lung cancer, and so that was a basic understanding of how the immune system worked, led to that development of drugs. But then we got a better understanding of what the immune system was actually seeing on the tumor cell. And it was seeing proteins that had mutated, right? Which made a lot of sense from an immunologic standpoint. And then the huge breakthrough came that showed that a particular abnormality in the cancer cells, microsatellite instability, actually caused these mutations, and tumor cells that had microsatellite instabilities were much more susceptible to checkpoint inhibitors. Now, the breakthrough was that then all of a sudden those checkpoint inhibitors were approved for cancers with MSI or MSI that were microsatellite unstable. Nobody had actually tested the drug in all these cancers, right? They just said, this is the biology and it works in these cancers. So the FDA is just going to approve for 30 other cancers. I don't remember how many it was. It was a lot, that there were no clinical trials done. They just said, the biology is here. The drug should work. We're going to approve it. So all of a sudden you have this enormous acceleration of clinical introduction that was based on our basic understanding of how this worked. And that's the key piece.
Fantastic. You know, you talked about these amazing advances, this acceleration of research, but in the end, fundamentally, it's people doing a lot of this work
Yes.
behind the scenes.
Yes.
So give me a sense if one, if things have changed from the Farber days, what a modern day lab looks like in terms of day-to-day personnel, what a principal investigator or leader of the research lab does, and how does that fit into the bigger theme of advancing science?
Well, it used to be because, we were dumber back when I started. There weren't that, the questions that we were asking were important, but they weren't particularly complicated. And so you, as a lab, could work on something just by yourself. Your laboratory technicians, your graduate students, maybe a lab of 8 people, could actually really by themselves tackle a question. The technology was relatively, I don't know, it was Stone Age compared to now. The approaches were simple. The questions that we could ask were relatively uncomplicated simply because we didn't have the technology to ask bigger and harder questions. Now, you still might have a lab of 8, but all the things that your lab has to do, either or learn how to do, has become much more complicated. So a lot of the new technology is sequencing. It used to be back when I did sequencing, which was a long time ago, you could only read 200 base pairs at a time. So it was 200 DNA, pairs. You could only read 200 of them at a time, and it would take you literally a month to sequence 1,000 base pairs, maybe part of one gene. Now you just put it in the next generation sequencer, it coughs out the entire genome in 3 hours, right? I mean, the technology has advanced so quickly. Now you have to figure out what you're going to do with all that data, right? And so what has happened is that science has actually gotten a lot more collaborative because we can't know everything, right? I can tell you how to do myeloma, but I can't tell you how to do, prostate cancer or the biology. So we have become a lot more collaborative across multiple institutions. And this is the great thing about science, is that for the most part, all scientists really love to share their work, their data. They like to go work with somebody that says, hey, I have a really interesting question, but I don't know, I don't have the technology to understand how to do that, or I don't have the patient samples to actually look at it. Let me go reach out to my friend who works at Emory, and say, Larry, you guys have been doing this. This is the question we want to ask. How would we do it? And can you help us? And that's become very much the case in science today, is that people have really started to collaborate together and bring in new technologies that maybe only one or two people know how to do, but they're very welcoming in sharing that expertise, and we go out and learn how to do that. So that's probably the biggest change. It's really gotten to be a much more communal effort than it was maybe 30 years ago.
Yeah, I'll just echo some of that sentiment. The technology has changed rapidly. One thing that's been great is the access to information has changed as well. So
Yep.
one time having to read a manual on how to use a flow cytometer is now a YouTube video on how to do it,
Yep.
which is fantastic. That's how I learned my first
Yep.
flow cytometer, how to do the
Well,
compensation.
you probably don't even remember having to go to the library and Xerox articles, right? I mean, there was no internet, right? You go to the library and you stand in front of the Xerox machine and you're Xeroxing articles.
Yeah, it comes with like a huge packet of information. You just throw it away and you YouTube it, and it kind of
Yeah.
it helps you out. You get a bunch of people from all over the world showing you different ways of doing it. And then likewise, I was doing something in my lab that had never been done before in my lab, and so they actually sent me to a lab in the same building nearby that had
Yep.
expertise in it, and they said they're gonna teach you how to do it, and then you bring it back and you teach all of us how to do it.
Yes.
So there's a fundamental component of your more junior lab personnel becoming the spearhead for new
Yep.
technologies and new pieces of knowledge that ultimately, when you're a senior, you then teach other people how
Yep.
to do it and so on. It becomes a building block for how the lab grows and
Yeah.
develops its
And that's
repertoire.
the great part of graduate students and students in your lab. They're the ones that they have no problem going to do something they've never done before because they haven't really done anything before, and they're fearless. And they're the ones that go out and say, there's this really cool technology. The NIH is doing it in Washington. I would like to go learn, and you send them, right? Because they are excited to learn new things and bring that knowledge back to all of us.
And we feel important. Like when we come back, someone's asking us a question. We feel like
Yeah.
we're finally taking the lead. We get to help,
Yep.
colleagues out. They taught us a bunch of things, and now we get to return the favor. It's a very nice.
Part of
Uh,
your professional growth, right?
Yeah, yeah, it feels great. So in that vein, give me an example of a time where you've had a really technically challenging aspect in the lab, and how did you navigate that?
Usually, so I'm trying to think of what is the most technically challenging aspect in the lab we've had.
Or another question along the same lines, have you ever had results that were unexpected, and how did you navigate that situation of unexpected results or
Oh,
unexpected
well, that the
technical?
latter is all the time,
Yeah.
right? And the great discoveries are accidental. And I'll give you a specific example of this. So in the body, there are actually proteins that keep your cells either alive or cause the cells to commit suicide. And this is one of these. It's called apoptosis. It's your cell sensing, hey, something's screwed up here. And instead of allowing, my cell to turn cancers, I'm just going to commit suicide and die. And this is a normal process. Actually, it's a normal process in development. All of us are born with web fingers and web toes. And the reason that the webbing goes away is an apoptotic process that causes all those cells in the webbing to disappear. So way back when I was sequencing 200 base pairs at a time, we had a project that actually was trying to clone out the second pro or anti-apoptotic gene. This was a gene that actually prevents cells from committing suicide. Turns out that these are very important for normal biology, but also cancer cells use them to prevent themselves from dying. And so the second gene, we actually had the sequence from the first gene. This was from another group, and then we went looking for it. Actually, we went looking for it in the chicken genome. That's a long story that I'll tell over a beer sometime. But we went looking for it and came out with a gene. And we were reviewing it at lab meeting, and my lab chief, Craig Thompson at the time, was looking at it. And the person that did this said, well, the front end looks like the gene that has been another gene that's been previously described, but then all the sequence gets screwed up and it doesn't look anything like that original gene. And she said, I think this is just an artifact. So Craig, one of the great things that Craig did was he was very, very objective, and he didn't let what he thought was going to happen get in the way of his interpreting of what was actually happening. So he looks at this gene and his gene and it says, the gene remains in frame, which means from a layperson, genes encode proteins. And this gene still was making a protein, even in the part that had nothing to do with the previous gene, it still was making a protein. So that said to him that this was a real gene. And that ended up being the second anti-apoptotic gene that was ever cloned out. And that was because, yes, we expect something, but the best results often happen because something happens that we didn't expect, and we just don't say, well, it wasn't what I expected, I'm just going to throw that out. That's the important piece. It sounds like it's a simple thing, but all of us are wedded to our ideas. I make a prediction. This is how it's going to work, right? And then when it doesn't work, I'm disappointed. And then a lot of that disappointment means I'm just, that didn't work, and I'm not going to think about it. Instead of going back and saying, well, what is this really trying to tell me? And is there a great discovery in there? So that's an example, and one of a billion, right? That were actually, what did Pasteur said that luck favors the prepared mind. So if you're willing to see beyond what you thought was going to happen, sometimes there's some incredible magic in there.
Yeah, I also trained in a lab that was my PI was trained by Craig Thompson as well. I think that was a fundamental approach of objectivity,
Yeah.
but even more so that we remain curious and open-minded to what those things could look like. And then we kind of let it lead us, and then we sort of learn from that and then make better hypotheses going forward.
Right, right.
So I'm gonna transition a little bit. I'm gonna shift over into the clinical research space. And you described in great detail about how we move from a first in human early phase drug all the way up to FDA registration and getting the drug approved. But for a patient, what does that experience look like from the different aspects of clinical trial development, from phase one all the way up until phase three?
So from a patient standpoint, I mean, I would say that has actually become blurrier over the last 10 years. It used to be that, and we still say this for this drug has never been tested in humans. It's a phase one study, and our objective is to actually to understand, is it safe to give to people? And we still tell all our patients that are going into a phase one study that there's no guarantee that this will do anything to your cancer. We believe it will based on the translational research and all the research that we did in the lab, but we don't know because we don't have any evidence. So we tell patients that, all right, this is something that we don't know if it's going to work, and our primary objective is to see if it's safe to give. So if it's safe to give, then we can go to the next phase and try to figure out, is it actually killing cancer or not? Now, I have to really give a shout out to all the patients out there. All of our patients have been so willing to be part of clinical trials. And the number one thing I always hear, especially for these early phase clinical trials, is a patient will say, well, if it doesn't help me, it might help somebody else. And that is so heartwarming. And that's why we do what we do. So phase one really is to understand how, is this safe, what to expect from it. Now, I would say that for example, in one example is with the immunotherapies, especially with the chimeric antigen receptor T cells, the CAR T cells, the phase one studies to look at toxicity also gave this enormous it kills cancer signal. And so that phase one rapidly accelerated into a phase two. Phase two is trying to figure out what's the best way to give it so we get the maximum amount of, cancer killing. And so those studies actually are beginning to blur together. But phase two is really, hey, is this working? And then phase three, which are less and less, although they're still there, is there actually ask, is it working better than our current therapy? One of the hardest things for phase 3 studies is that if one of the groups that is getting treated in the phase 3 study, or an arm, as we call that, is actually getting a placebo. Because it turns out that nobody wants to get the placebo. I mean, people are going into the clinical trials hoping that this is going to do something. They don't want to be the comparison group. So that's something that's a little bit harder to describe. But I would say that one of the things uh, um, that is true is that the clinical trial experience has gotten more and more hopeful. I think that because the technology is advanced, the rapidity by which we can do the studies that is advanced really has been, all right, we're just not just doing an experiment on you. This actually has real chances of being effective. So I think that that's one of the more exciting things over the last 10 years that really has helped us. And then our clinical trials have gotten much more sophisticated. So we're actually, instead of just looking at, hey, did the tumors get bigger or smaller? When we gave you this drug, we're doing a lot more of the analysis using these high-tech approaches to read out things that we may not have been able to tell, just looking at whether or not tumors get bigger or smaller when you give them a drug. And I'll give you one example of why that made the difference. One of the other seminal trials that actually changed the way we think about immunotherapy was one of the first cellular immunotherapy trials using a kind of cell called a dendritic cell to activate your immune system, and it was done in prostate cancer. So men that had gotten all the therapy, they had nothing left, were eligible for this trial, and this essentially is a vaccine. It's a live vaccine. They got this vaccine, and when you did the initial analysis, none of their tumors got any smaller, right? There was no evidence that this was actually making the tumors shrink. However, when you did the secondary analysis, those patients that got the Dendreon vaccine actually lived longer, significantly longer than the people that didn't get the vaccine. And then they went back and said, well, tumors didn't get any smaller. It's sort of that traditional framework we think about for therapies. Traditionally, the tumors didn't get any smaller. What's going on? So they went back and looked, and they actually found that the tumors grew slower. They didn't stop growing. They didn't get any smaller, but they grew slower. And what kills a patient is that tumors get bigger. If the tumors actually stop growing or grow a lot slower, it takes much longer time for that tumor to become a problem. So that was the first time that anybody really in a clinical trial in an immunotherapy clinical trial says, we are measuring the wrong thing. We have been measuring whether or not your tumor gets smaller. That was the wrong thing to measure. What we should be measuring is whether or not your tumor is growing slower. And that was an enormous paradigm shift. And it turns out that there are probably lots of immunotherapy trials that were, in fact, successful, but people were measuring the wrong thing. So the evidence was there. We were just looking in the wrong direction.
Yeah, I'll just add and kind of to summarize that, good science in the clinical and translational aspects ultimately makes for better clinical trials. But even so, a lot of the clinical trials that we've done have ultimately informed the basic and translational components that are
Yep,
going on at
goes
the same
backwards.
time.
Yep,
We go
yep.
kind of go a little bit backwards, figure out that a drug is not working in this very broad population, but then all of a sudden we see, hey, in this one particular group of patients like EGFR, we
Yep.
saw that patients who had EGFR mutations were doing exceptionally well. And so then we kind of retooled how we did the clinical trial. We realized that those patients were the ones that were getting the major benefit from these drugs. And ultimately, we have an entire field dedicated to that research now, focused on EGFR mutations.
Right.
I also add that, one of the things that we've done with statistics is we've worked really hard in making sure that if, the study is looking to have 100 patients on the study before it looks at the data, then now we have these adaptive trial designs where we can look intermittently through the course of the study. And if we find, that the drug is working exceptionally well, we sort of move on. And
Right,
likewise, if we find that the drug is not working well enough, that we don't put patients through an
right,
unnecessary clinical trial. And so that's really where the science and the math have sort of come
right,
into play and made for a better clinical trial.
right, right.
Okay. I'm gonna transition over to some of the questions that we received prior to this, as well as some of the questions that are ongoing on the chat. But the first question is for Dr. Lee. If Dr. Lee could have one cancer research question answered, what would it be? And what do you think will be the big discoveries in the coming years?
Some people have heard me say this, but the holy grail is that if we can prevent you from getting cancer, we've cured it, right? You don't even get it. So one of the big questions is that it turns out that a lot of cancers that people get have a precancerous stage. This is turning out to be in lots of different cancers, multiple myeloma, some of the leukemias. Actually, cancer appears to have a lot of these things early on that aren't cancer, but are going to, in some people, will become cancer. And so the real question is that, and I suspect that almost all cancers have a precancerous stage that probably exists for years before it becomes cancerous in a population, a subset of people. I think one of the big questions is why does that happen? Why do these precancerous lesions or abnormalities in 2% of the population become pancreatic cancer, where the other 98% of people never get anything, even though they have the same number of these pre-malignant or precancerous lesions? Why is that? Because if we could actually prevent that from happening, which is a concept that is called cancer interception, if we can prevent that from happening, then we can actually prevent people from getting cancer. And that would be a huge win because then we don't have to treat people that actually have cancer. So if I were to say that if that one thing, now it's probably going to be, the reason I paused a little bit is probably going to be 75,000 things that are involved in preventing that from happening. But it's there. I mean, I think that we would not have evolved as a species if we would get cancer at a drop of a hat. So there are probably lots of mechanisms in play to prevent that from happening that just break down in a small number of people that are unfortunately go on to get cancer. But I think understanding that, be able to screen that and be able to manipulate that or prevent, that cancer from progressing is going to be a huge breakthrough.
I'll just make a quick plug towards the other aspect, which is cancer screening
Yes,
and curative intent treatments that are available for earlier diagnosed
yes,
cancers have a huge impact on overall survival. So
yes,
if you are eligible for cancer screening, we also encourage that. But certainly prevention is the
yes,
topmost
yes.
priority and the holy grail for cancer related issues.
Right.
This is going to be a two-part question, but I think one that's very important, especially in the times that we are living in. Why is cancer research so expensive, and why does it take so long before we see a product come through?
Well, that's a great question. There are lots of actually pieces to it, but if you think about, all right, cancer or research in general, cancer research, biomedical research is essentially equipping a ship to go out and on an expedition into a space that or a place that nobody's ever been before. So we have no idea what to expect. So a lot of the technologies that we use are very bespoke. They are very sophisticated. But they can tackle lots of different things. So it's not like, hey, I've got a widget that does one thing. I have this technology that has to do 270 things because we don't know what we're going to need once we're out, exploring. So part of it's that, and part of it is that a lot of the reagents, for example, are expensive. You can't go to Walmart and buy them. They're not mass manufactured. They can be very niche. So that drives up the expense. I would say that the clinical trial expenses are probably what a lot of people think about when they say, "Hey, why is research so expensive?" And an important reason is that So if you're going to ask a person, a patient, to enroll on a clinical trial, let's say a phase 1 clinical trial, it is your moral and ethical responsibility to make sure that the data, the information you collect from that patient's participation is good, it's accurate, it's accurately recorded, it's logged in, and we can go analyze it. That requires an enormous amount of care, and right now, it requires an enormous amount of hands-on making sure that data is there, because it's unethical to enroll a patient on a clinical trial and then just not collect their data properly, right? That person is essentially consenting to risk their lives, and if you don't honor that by doing the best you can with the information you get, you are actually not doing the right thing. So a lot of clinical trials is very, it's costly because it has a lot of regulation, it has a lot of care and importance in understanding it. Now, that having been said, not everyone is concerned, but there's a realization that maybe some of our clinical trials are a lot more complicated than they need to be. And that's part of it's historical. We've always done it this way. We need to do these gigantic trials. And part of it was before we had better ways of analyzing. Hey, it used to be that I needed 500 patients to come to a conclusion. Now I need 30. And so a clinical trial of 30 people using sophisticated analysis is a lot cheaper than having 500 people and using Stone Age analysis. So there is a movement to make those trials less expensive. Certainly, other countries have figured out how to do that also, which is one of the challenges that we have in the U.S. is to how do we actually implement those. So it is expensive. It is expensive because, I mean, in the terms of developing a therapeutic, a treatment, a drug, everybody who's going to get that drug once the FDA is approved is relying on the data that was generated in the clinical trials to be accurate, right? And well done, because the last thing you want to do is approve a faulty drug or any kind of faulty anything that is going to go into millions of people, right? So there's a lot of care, and care costs money.
I will say I might be personally responsible for some of those expenditures. In my first year as a researcher, I went through two DNA extraction kits and failed to use them well, and it was like $2,000 for the lab over the
Uh,
course of a day and a half. So my postdoc was visibly frustrated with the amount of time I wasted in the course of a day and a half.
yeah,
But it is an iterative process. It does take a lot of time and a lot of money in terms of
yeah,
reagents, and then the
yeah.
clinical trial processes also.
And the other thing is that my mentor Craig Thompson said, the difference between Nobel laureates and the rest of us schmoes doing research is that Nobel laureates have 20% of their research work, and the rest of us have 10% of our research work, which means that Nobel laureates have 80% of what they do fails, right? So you have a failure rate of 80%, right? You're going to spend a lot of money and not generate anything because your experiments, you guessed wrong, or this wasn't what, the process was, the mechanisms. So there's an enormously high failure rate, even with the most successful of scientists, that adds to the cost of doing the research.
Absolutely. I'll sort of end this with one last question. I think it's a really important one. What advice would you give to a student or young professional who wants to work in cancer research but doesn't know where to start?
So I think that there are multiple parts to that. The short answer is start anywhere. Because as you've heard from both Dr. Maniar and myself, how we got into research was not where we ended up, right? We started somewhere else and we got excited about the process and the importance of what we're doing, and that will guide you in the right direction. I think that so just start anywhere, go to a lab, go to a summer research program, and just see what that's like. Work in a clinical trials office to see what clinical trials are like. Do those things just to get some sense of what is important. Now, I think that there are two other key pieces to know, or to do this for the long term. One is that you have to believe that what you're doing makes a difference, right? And so we always say in the Cancer Center, we have lives to save. That's why we do research. We don't do research because, we're going to get famous or, I'm going to end up on Oprah or, somebody's going to show up with a check for $1 billion, right? Or, none of that. I do it because well, you have lives to save. And I see that every day in my clinic. And I think to myself, there must be something better than what we're doing right now. So one of them is that emotional connection. So everybody thinks that science is science and is dry and analytical. No, the key pieces are what makes a successful scientist is they're passionate about their work because they believe it's going to fundamentally change the world. And that's important. The second piece, and I always say, somebody said, how do you know if research is right for me? And I say, well, how do you know if anything is right for you, any profession or whatever? I said, probably the easiest way to tell is, are you having fun when you do it? Because fun might sound like a trivial thing, but there are pieces of fun that are critical for you to be successful. One, you're having fun because you're good at it, right? I might want to play in the NBA, but I suspect that I will have no fun playing in the NBA because I'm terrible at basketball. That was an admission to all our Hoosiers out there. So one, you're having fun because you're good at it, or you have talent in it. Two, you're having fun because it engages you emotionally. This is fun. This is exciting. This is really cool. It sparks your curiosity. And I think that those things are key as a simple test of whether or not research is for you or science for you or whatever, is that when you do it, were you having fun? You walk home and say, wow, that was the coolest thing ever. That's a sign. That's a great litmus test of whether or not this is for you.
Yeah, I'll say I knew it was right for me when I would go home after a really long experiment and I would dream about the experiment.
Yes.
I would
Yes.
dream about the biology underlying what I saw and I would
Yep.
think about it and I'd wake up and I'd be like, I think I know what's going on. And I would just go
Yes.
and analyze the data. And I'm like, nope, I didn't know what's going on.
Yeah.
Back to it and I'd try again.
Exactly.
And I would start dreaming about it. And I was like,
Yeah.
I think this is very much a science, right? This is iterative, there's processes, there's recipes, but there's an art form to it. And it's an
Yes.
art that you get over time and exposure
Yeah.
and through seeing how other people do it.
Yeah.
And then your data is your own data, and you get to help
Yes.
define how that looks and what the
Yeah.
story of the biology goes with
Yes.
it. So
This is your tale.
this is your tale. Yeah.
Yeah.
Okay, well, thank you so much. Unfortunately, we are running out of time. And thank you to all those who joined us this month for Simon Says and all the great questions that were posted. And a big thank you to Dr. Lee for providing incredible insights on the topic of cancer research. We hope to see you at a future Cancer Center event. You can find a recording of today's session and information about upcoming sessions at cancer.iu.edu/Simon Says. Have a great day.
Great. Thank you all.